Nicholas is working with leaders in both industry and academia to support their research needs. Nicholas is part of the team that helps to guide in the use and the development of recombinant proteins that can be used for COVID-19 CAR T-cell therapy, as well as many other immune and autoimmune conditions. As a member of ACRO, he is highly interested in leveraging the company's resources and the state-of-the-art technology to help their partners and the clients develop next generation therapies. Now I'm going to turn it over to Dr. Abuid for the presentation. Thank you.
Thank you for that very nice introduction. And yes, as mentioned, my name is Nicholas Abuid. I'm an application scientist for ACROBiosystems. We're the leading manufacturer of recombinant proteins for a lot of different applications, including ADC, bispecific antibodies, and recently for the COVID-19. But today I really want to talk to you about how we've engineered bispecific antibody targets as a novel arsenal in immune therapy. Before I proceed to my presentation, I would like to give an outline of the content in which I will start talking about bispecific antibody backgrounds and a format and trend, followed by our journey on bispecific antibody development, and finally some of the featured products and technologies we've been working on. So with that, I'd like to proceed in introducing bispecific antibodies. This refers to a large family of molecules that recognize two different epitopes or antigen, and this dual target functionality led to an increase in interest in this for their therapeutic applications due to the potential of bispecific antibodies in precise targeting and synergistic therapy, which makes it stand among antibody therapies due to their broad prospects in immunotherapy. So there's a few advantages of bispecific antibodies that I want to point out. First, this two antigen binding sites can be used to bind tumor cells and immune cells, as well as redirect and recruit these immune cells to the surrounding tumor cells and enhance cytotoxicity. There's also this simultaneous blockade of two different signaling pathways that result in enhanced cytotoxicity, but also mitigate this drug resistance caused by tumor pathway activation. And finally, after binding to two different cell surface antigens, there's a potential increase in binding specificity, but it can also bind to two epitopes of the same receptor and target the tumor growth environment and mitigate some of the side effects caused by the off-target. Following, I would like to talk to you about some of the different roles of bispecific antibodies. So we can start talking about bispecific antibodies and how they bridge two or more cells, binding the antigens in a particular cell. The most famous one that most of you know is T-cell engagers, directed toward cancer cells. Then you have bispecific antibodies bridging receptors designed to bind multiple receptors with the same cell and allowing a higher affinity binding via proximity and a high concentration effect. And yes, in addition to this, there's also bispecific antibody activity cannot be mimicked by combination of two parental antibody, and therefore there's this obligate bispecific antibody with unique bioactivity profile. Another bispecific antibody role is cofactor mimetics that binds enzymes and substrates, creating this complex. And this is seen as a desirable feature that binds to different epitopes of the same antigens to increase the avidity. And there's also this piggyback bispecific antibodies, where one arm specificity of an antigen is leveraged for a second arm to be highly specific to the neighboring intended target. And this agonist bispecific antibodies activate downstream receptor signaling and binds multi-component receptors with simultaneous binding of the complex. Depending on the disease biology, mechanism, and biochemical consideration, bispecific antibodies can be engineered to function as required. I would like to very briefly talk to you about some of the formats of bispecific antigens. I'm just going to breeze through this in the interest of time. There's IgG-like and non-IgG-like. IgG-like has several advantages, such as is very similar to natural antibodies, is Fc-mediated effector functions, long half-life, and we develop very mature process suitable for purification production. However, there's poor permeability and a complex structure. The non-IgG bispecific antibodies can be better in tumor penetration and have fewer side effects. However, it does have a short half-life. There's a trend going on since the 1960s, where the first bispecific antibody was generated using this thiol redox method to couple FAB regions of two antibodies within different specificities. But later on, with the advent of hybridoma technology by Kohler and Milstein in 1975, there was this antibody revolution that was followed by 2 decades of development of antibody engineering. And we came up with methods such as single chain variable fragments and solutions to asymmetric antibody pairing. At the same time, there was this 30-year effort in T-cell redirection for therapeutic. This culminated in 2009 with the approval of catumaxumab, and which was used for patients with malignant asites. However, this was later withdrawn. But either way, there was four total commercialized bispecific antibodies, commercially known as Blincyto, Hemlibra, and Rybrevant, used for acute lymphoblastic leukemia, hemophilia, and non-small cell lung cancer. In addition to this, there's over 85 bispecific antibodies in clinical trials, and we've really seen how fruitful these advances have been, with commercial pipelines that are considered from regulatory approval within the next 5 years. We've also seen an increasing number of licenses, industry and academia increasing the number of pipelines and studies on this, being used in different applications for oncology, autoimmune disease, neurology, and an increase in sales of bispecific antibodies, which was predicted to be exponential. The bispecific antibodies, however, do not exist in nature and need to be prepared by technical engineering methods. The development is very complex, and there's technical barriers, and we need to be careful in selecting the technical platforms and targets since they're very challenging. But ACROBiosystems adheres to the concept of where proteins and innovation advance in biomedicine, and we've been committed to develop a broad myriad of targets in bispecific antibody development. One that I really want to point out is CD3, which is a hot target for bispecific antibodies. And I would like to talk to you a little bit about some of its background. T-cell bispecific antibodies are engineered molecules that include binding sites to T-cell receptors and tumor-associated or tumor-specific antigens. The CD3 molecules has the function of stabilizing TCR structure and transmitting activation signal. These antibodies against CD3 molecules can stimulate or block T-cell activation signal transduction. and can help eliminate effector T cells or induce the production of regulatory cells. So CD3 molecules can bind non-covalent TCR non-covalently, and there's 4 different types of subtypes of CD3, including delta, epsilon, gamma, and zeta. There's also this complexes of heterogeneous heterodimers of delta and epsilon, and gamma and epsilon, and this targets can this targets they complex through the alpha beta chains of the TCR, and their CD3 targeted therapeutic antibodies usually recognizes CD3 epsilon in the heterodimer complex. However, CD3 epsilon and delta can randomly form incorrect heterogeneous complexes, which may lead to a loss of activity and high batch consistency. So for that reason, to accelerate the research and development of bispecific antibodies, and ACROBiosystems has developed a series of homogeneous CD3 epsilon, delta and epsilon and gamma and epsilon proteins with very high bioactivity, which were verified by SDSPage, non-reductive SDSPage, and MALS. We have a long list of epsilon and delta, about 18 products from different species and tags. Also, we have around 13 products of CD3 epsilon and gamma of different species of human, mouse, cyno, rat, rabbit, and other different tags, as well as just the single proteins, CD3 epsilon, delta, and gamma, to ensure that our collaborators and our customers have the highest level of products and don't have to really worry about consistency and variability from batch to batch. We do provide this SDSPage and SEC MAL reports with very high purity and consistency to ensure that you're getting this very consistent batch to batch and low variability. In addition, for this, we also have Bioactivity ELISA, SPR, and BLI that we provide not only these reports but also the protocols to help facilitate your studies and give information on concentration, different buffers, different information that will help you make sure that your experiments are run smoothly. Finally, we also promise high batch to batch consistency, as shown by ELISA, and we have a report that it was showing bioactivity comparing different lots, but if for any case you do want one specific lot, we are more than happy to share to hold this lot for you and for future experiments. So in conclusion, bispecific antibodies have come a long way from antibody engineering challenges to becoming this key arsenal against malignancies. There is this increasing number of commercial pipelines, interesting commercial pipelines, and we're excited to be part and help assist these studies. And there is also work in addressing the biological complexity of diseases. And we really hope to be a part in your experiments and assist your studies when realizing these bispecific antibody studies. However, we do go beyond CD3 proteins to FC receptors for IgG-like bispecific antibody development. We also provide services via BioAccord and Octet. We also, in addition to this, provide complementary molecule interaction analysis services, antibody screening, characterization, consistency evaluation, etc. And finally, just want to provide ACRO's contact information and some of the other products and services that we provide beyond antibody targets, immune checkpoints, not only for immune oncology and autoimmune disease, but also for viral proteins, and really help. We're really committed to being part of your experiments and try to customize any products or any proteins to assist your pipeline goals. And with this, I just want to thank you so much for your attention, and would like to open the floor for some questions.
Thank you so much, Nicholas, for your great presentation.